afternoon, everyone. My name is Halyn from Seoul Foreign School. Today I'm excited to share our significant research findings on the integrated effects of probiotics on adolescents' emotional, cognitive, and neuronal functions via the gut-brain axis. In contemporary society, adolescent mental health is a critical concern, and our study offers a novel approach to address this growing challenge. The gut-brain axis represents a bidirectional communication network between the gastrointestinal tract and the central nervous system. This revolutionary concept postulates that the gut microbiota profoundly influences brain function through various mechanisms, including the synthesis of neurotransmitter precursors, the production of essential metabolites like short-chain fatty acids, and the modulation of immune responses. Essentially, the health of our gut is intricately linked to the health of our brain and our overall mental well-being. Previous studies have largely focused on animal models or isolated molecular pathways, often failing to integrate clinical outcomes with mechanistic insights. To overcome these limitations, our research adopts a multi-layered design. We have integrated human randomized controlled trials with parallel cellular mechanistic studies. This innovative approach allowed us to simultaneously gather both clinical evidence from human surveys and molecular mechanistic evidence from cell models, providing a more comprehensive understanding of probiotic effects. Our study was guided by three core hypotheses. First, we hypothesized that a 2-week probiotic intake would enhance emotional well-being and cognitive function in healthy adolescents. Second, we proposed that probiotic-derived factors would strengthen the integrity of the intestinal epithelial barrier. And third, we predicted that gut-derived signals would activate neuroplasticity-related signaling pathways in neuronal cells. To test these hypotheses, we conducted a 14-day randomized controlled trial involving healthy adolescents aged 15 to 18 years old. Participants were randomly assigned to either a probiotic consumption group or a control group. We assessed a range of outcome measures, including the WHO Wellbeing Index, PHQ-2 Depression Scale, Visual Analog Scale for Stress, as well as cognitive performance tasks like the Stroop task and Tubac task. We also monitored gastrointestinal symptoms and sleep quality. Our human trial yielded promising results. The probiotic group demonstrated significant improvements across key indicators of mental health and cognitive function compared to the control group. Specifically, we observed a notable increase in emotional stability, reflected in improved well-being scores and reduced depressive symptoms. Furthermore, cognitive performance, particularly in attention and working memory tasks, showed significant enhancement. These findings suggest that probiotic intake can meaningfully contribute to the emotional and cognitive health of adolescents. Additionally, gastrointestinal discomfort, including bloating and abdominal pain, was modestly alleviated. Collectively, these findings indicate that even short-term probiotic intake can meaningfully enhance emotional stability and cognitive function in adolescents. To delve deeper into the biological underpinnings of these observed improvements, we established an integrated in vitro model. We first prepared cell-free supernatants from the probiotic beverage. These supernatants were then applied to a COCO2 intestinal epithelial cell model to mimic gut absorption. Subsequently, the conditioned media collected from the basolateral chamber of the COCO2 model, representing signals transmitted across the intestinal barrier, were applied to the SHSY5Y neuroblastoma cells, simulating neuronal responses. Our in vitro experiments revealed that probiotic-derived factors significantly enhanced intestinal barrier integrity. We observed a robust increase in transepithelial electrical resistance, or TEER, values in the probiotic-treated COCO2 monolayers, indicating tightened cell junctions. TEER is a measurement technique used to quantify the integrity and permeability of epithelial or endothelial cell monolayers. A higher TEER value indicates better tightness of the cellular barrier, suggesting effective resistance to ion passage and a more intact barrier function. FITC dextrin utilizes fluorescently labeled dextrin, commonly 4-KDA, to assess paracellular permeability, which is the passage of molecules through the spaces between cells. Increased flux of FITC dextrin indicates compromised barrier function, often due to impaired tight junctions. Our results showed a significant decrease in FITC dextrin permeability, suggesting that the barrier was strengthened after probiotic treatment. Immunofluorescence staining further confirmed these findings, showing clearer and more continuous localization of tight junction proteins like ZO1 and Claudin-1 along the cell junctions, collectively indicating a strengthened and more organized intestinal barrier. Moving to the neuronal component, we found that conditioned media from probiotic-related cockatoo cells significantly modulated neuronal function. RNA sequencing revealed a marked upregulation of genes associated with neuroplasticity, including pathways involved in neuron projection development, synapse assembly, and axon guidance. Critically, RT-qPCR analysis validated a significant increase in brain-derived neurotrophic factor, BDNF, mRNA expression. BDNF is a key mediator of neuronal plasticity, crucial for neuronal survival, growth, and synaptic function. These results strongly suggest that gut-derived signals promote neuroplasticity in the brain. In conclusion, our study provides compelling evidence that probiotic consumption can positively impact adolescent mental health by reinforcing the intestinal epithelial barrier and promoting neuronal plasticity via the gut-brain axis. This offers an accessible and cost-effective strategy for mental health promotion. By integrating probiotic-enriched foods into school meal programs alongside education on sleep hygiene and stress management, we can establish comprehensive school-based mental health initiatives. Our research provides a crucial scientific foundation for these programs. While further research is needed to identify specific active compounds and optimize probiotic formulations, our findings underscore the immense potential of nutritional interventions in fostering adolescent well-being. Thank you.